Thank you so much. Thank you for having me today. Thank you for organizing this event. It actually does a very important thing, is to disseminate the information and knowledge about the microbial applications. Quite a lot of people are well aware of mammalian cell cultures and how to process them, how to develop new processes, and when switching to microbial, they bump into a lot of obstacles because bacteria are different. And the main difference is the intensity of the processes. And you have to adapt to much higher intensity of the processes. And actually, my talk today is how to approach the development of a robust microbial bioprocess, taking into account, considering this intensity. A very important disclosure, as usual, standard. A very humble slide about myself. Who am I, what I'm doing, with my address, website, and actually at the moment I am a biotech consultant and I'm about to start hopefully my own company within the next year. That's why I'm proud to call myself entrepreneur. Before that I led a small research facility specialized in bioprocess and analytical development at Oxford University, more specifically Jenner Institute. Therefore, I am aware of malaria vaccine R21 matrix, which was mentioned today during the first talk. Before that, I led process development at Ipsen, and before that, I worked as a head of biotechnological laboratory in a company, ContiPro, who is the main producer of hyaluronic acid on European market. So if and when you apply your hyaluron-based cream on your face, highly likely I had a hand in developing this bioprocess that led to your cream. So whenever we speak about the robust bioprocess, we can refer to ICHQ8, which says that the robustness is the ability of a process to tolerate the variability of materials and changes of the process and equipment without the negative impact on the quality. In other words, the process should be resilient to variability and can handle slight variations in input parameters like pH, temperature, etc., etc. It all must result in the same consistent output. So regardless of all the small variations, you should have the same quality and quantity of the material you require. And to achieve this, you would obviously use defined operating ranges. For example, for pH, it won't be pH 7, but for a robust process, it delivers the same result within the pH range of 6.8 to 7.2. And you would obviously use these process parameters within the operating range, and also raw material specifications would be also within the specific range. For example, you can use glycerol to feed your culture, and the glycerol range should be 90 to 98%, and it doesn't really affect your process because it's robust. And also, if your process is robust, it's much simpler to scale it up from smaller volume to higher volume to address the needs either of your customer, if you're CDMO, or your own needs, or the needs of the patients. Because here, whenever we produce any kind of biopharmaceutics, we at least must, in theory, think about the patients. And I hope we do. So altogether, whenever we speak about the robust process, we speak about control. We really want to control everything. And I'm not saying micromanaging people below you. We are controlling the bioprocess as tightly as possible. So one of the, let's say, tools how to increase the control of the bioprocess is to control the complexity of the bioprocess. In the center of any bioprocess lays a living organism. A living organism, by its nature, is very complex. Even the bacteria, which is fairly simple compared to humans, it's much more complex compared to organic synthesis reactor, where everything is defined And if we reduce the input complexities that go into your living organism, into your host, we would decrease the complexity in input. And we should not be stressed too much about deviations, variability, uncertainties, and other risks associated with high complexity products. How we can achieve this? First of all, we can look at the host. And I recommend, when and if you can, switch to simpler host. In today's topic, obviously, the simpler host will be probably the bacteria, or maybe yeasts. Mammalian cell cultures are much more complex. They have more genes, they have more complex metabolism, and they bring a lot of complexity into the process. Some people also mutagenize hell out of the host to reduce its genome even more, and sometimes to reduce the genes that produce specific contaminants that make downstream process more difficult. To be honest, I think it's a very difficult way to follow, just because you don't know what would happen within the cell if you break one gene. You remove the protease, it might break something else, and this protease is indeed essential for bacterial strains. So usually it's all limited to removing some non-essential proteases that might damage your protein, removing some nucleases that might damage, for example, your plasmids, and nothing more. The rest is quite risky. So personally, I wouldn't recommend to go too deep into host mutagenesis, especially if you don't have enough resources for multi-parallel screening, or if you don't really know what you do. The other topic that brings complexities is input raw materials. Here we can compare a complex medium that is composed of yeast extract, some kind of peptone, some salts, some sugars, and comparing it to minimal medium that is capable of maintaining the metabolism of the cell of your host. And obviously, switching to minimal defined medium would decrease the complexity, because all the complex media are made of, again, biological input materials, and you never know what happens inside this production. Also, additional source of input complexity is external environment. What's the temperature, what's the pH, what's the pressure, what's the moon phase, etc., etc. And to actually circumnavigate about this complexity, you would rather put a lot of sensors for monitoring all the parameters you can. I am quite an adept of process analytical technology, or PAT. And one of the best tools here would be probably Raman spectroscopy, where one sensor could give you quite a lot of information about the processes that are happening in the medium during the fermentation or even during downstream processes. And also, all the sensors are not just for decoration. They inform you to keep the parameters within the specified ranges so that we reduce the complexity, we reduce the variabilities at the input. And also, the last source of complexity is ourselves. Due to our human nature, we do mistakes, even if it's not intentional, especially when it's not intentional. And especially risky this is for more experienced people, because when you feel experienced, you don't care as much and you don't follow the protocols, because what they know, what I know, obviously I'm smarter, and you can't avoid, forget some certain things, switch it on, switch it off, etc. And there are two approaches. One is so-called GMP approach, where you have two operators and one witness, and you tell the operator to do thing, you write it down, the witness sign it off, and you have like a triple control of our own human nature. to reduce these risks. Obviously, it's costly, it's very slow, but if you worked in a GMP environment at least once, you know this is fairly common, and it's exhausting, especially when you are suited in the whole garment, which is not really breathable. So how we can tackle it? I like automation. The automation removes the human from the process and removes this source of variability and complexity. So do your best always to try to minimize human input, and automation is one of the ways how to do it, especially now when there are quite a lot of systems that are CFR 21 compliant, and they are being used to replace human input in GMP processes. And regulatory bodies are fine with you using this software. So, as you might have guessed, I quite like automation because it controls environment, it reduces the human factor, and also I quite like chemically defined minimal medium, just because when you don't have a lot of input into the cell, the number of metabolic pathways is limited, so the cells behave better, they are more predictable, just because of tighter control of the cell metabolism, and also you have less in input, it's easier to control your logistics, and your supply chain is also very easy to control. I remember when we worked on one bioprocess, we had to test different ester extracts to find the right one. We had to test different peptones that are non-animal derived, because initially the process was animal derived. And it took some time, and it wasn't just to switch it all and to identify the suppliers. It was also very important to assure these suppliers exist and produce this product at this level of quality over the next 10 years, or whatever your cycle, life cycle of the product is. So by having chemically defined medium, it's just a tiny bit of salts, and it's very easy to source from different sources, GMP quality, any quality you require. So about the automation. I remember when I was invited to redo the automated process. So the automated process was indeed sort of automated, but to set it up, I had to do approximately 50 mouse clicks per bioreactor, 13 different values to type in from the keyboard, and times 12 because it was a multi-parallel 12-fold system. So imagine that just sitting there, typing it all in software when your bioreactors are already switched on and ready to run. It took, for me, as being a quick typer, approximately an hour. For my student, it was closer to 2.5 hours, just to double check whether everything is entered correctly and so forth. Still, 1 or 2 fermenters out of 12-fold run failed, just because, oh, actually, I didn't press OK there in one of the windows. And, but it was able to maintain the DO, etc., on its own. Still, I saw that it could be improved, and actually, this is an example of the same process using different code, no-code, and low-code systems that are being used in the industry right now. Sartorius, Eppendorf, web-based automation for anything that doesn't have any control software, actually. And I actually developed a script, let's say, let's call it this way, that reduced this time to 2 seconds per bioreactor, because it comes with all the variables embedded, and you could just copy-paste it all, or you could apply it straight to all bioreactors at once. And there is no room for errors, because it's applied automatically, and you don't have to press any 50 buttons. And this is actually the comparison of the normal DO control and script-driven DO control. So as you could see, the classic cascade-driven control without any additional fine-tunings wasn't really able to maintain this set 30% of DO, and the oscillation during the feed phase was not ideal. When the automated script was used, as you could see, everything improved. The oscillation is much lower here, and it sort of reflects the drops of the feed that go into the medium, and here it's controlled way better as well. So apart actually from reducing the mistakes, using novel algorithms, you could assure much more consistent run-to-run set of parameters. And now let's go to the comparison of complex medium and mineral medium. These are two overlaid fermentations, one with the complex medium, the light gray, and the defined medium is dark gray. And this is actually dissolved oxygen consumption, you can say. So at the beginning, the complex medium consumes, the cells growing in complex medium consume oxygen way faster because it's rich medium. They profit from having a lot of resources and they grow much faster than in complex media. They deplete it quickly, and when they deplete all the sources in the medium, you see quite a weird, strange dual oscillations up and down. That's because of the nature of complex medium. It has multiple sources of energy in form of different sugars, different amino acids, etc., etc. And when the main sugar that you put there, your glucose, is depleted, it switches to another source, and it gives this multiple peaks. And quite a lot of processes, they rely on this depletion of your carbon source detection to switch to the feeding. Here, you don't know when to switch. Usually what people do in the industry, they wait until the highest peak that sort of shows everything is depleted from complex medium. But during this period, the cells undergo multiple metabolic switches from one nutrient to another nutrient, to third nutrient, etc. It doesn't add a lot of robustness to your process. Also, you would notice by looking at the pH goes up and down, up and down. It jumps, as my Czech friends used to say, as a goat in Tatran mountains. So it's not stable. In the mineral medium, on the other hand, yes, they grow slower but steadily. And when your medium, when your carbon source is depleted, either glucose or glycerol, you see just one sharp peak. Because apart from this glycerol, they have nothing to eat. And you can say, yes, that's a right time to switch on, for example, your feeding. And this way it's way more controlled, way more predictable, and there aren't any metabolic switches in between. So yes, one of the reasons why you really should consider switching to mineral medium in general. The other thing, how to make a medium. For mineral medium, it's simple. You bought some salts, you mix them together, you dissolve them, you have your medium. This is just a tiny bit of the complex medium complexity. If you really want to produce the yeast extract that goes almost to every complex medium, you should use one of these methods, either physical, chemical, enzymatic method, how you would really produce it, and depending on the method, it is different. So you can't just take from a laboratory process the yeast extract and say, okay, I have this supplier of yeast extract, it should work. It may work, if you're lucky. It might not work, and you might want, you might need, unfortunately, you might not want, but you might need to test 15 different samples of yeast extracts. They might even come from the same company that produces yeast extracts, but they will differ. One would be chemically produced, one will be enzymatically digested, one would be chemically digested or physically digested, something like that. So again, the robustness and variability, robustness will be higher, variability of the mineral medium is much, much smaller compared to complex medium. And again, we really want to reduce the complexity of the bioprocess to ensure its robustness and reproducibility and stability and ease of scale-up. That's a very nice and colorful picture here that doesn't say much if you don't look at it for like 15 minutes. I'll try to reduce this time to 15 seconds. So here we have a comparison of the cells that are growing in mineral medium at relatively low growth rate, 0.22 mu, and rich medium, which grows roughly 4 times faster because it's rich medium. And this is a representation of all proteins being expressed in the cell. The bigger the tile, the more the protein. And there are like some groups of enzymes that are involved there. So in rich media, there is a certain shift towards glycolysis because it's a fast way to get cheaply a lot of energy without considering the accumulation of lactates or acetates, etc. It's because of abundance of anything else in this complex medium. And also there is a shift towards ribosome and protein synthesis because they already have all the amino acids in the medium and they can start producing proteins straight away with no problems. This assures very fast initial growth, and you could see it's 4 times higher, approximately. In mineral media, the shift is towards TCA cycle and transport. It means it's more robust and efficient in terms of utilizing the energy sources available in the medium. And the growth, yes, indeed, is slower, but it's constant. It doesn't fluctuate depending on the availability of thousands of components of complex media. Another very busy slide, but bear with me, it's not that complicated. Here we have like 3 main pictures. 2 of those are about rich media. This is rich medium in exponential phase. This is rich medium in stationary phase when it doesn't grow. Here we have defined medium in exponential phase. What we can see here, the main sick errors are towards TCA cycle and amino acid biosynthesis. Here, quite a lot of stuff goes into byproducts. Just because it doesn't go to TCA cycle, it's glycolysis and it's enough energy for these cells already. But when everything is depleted and it's stationary phase, it starts digesting byproducts via TCA cycle to have some energy instead, which you've seen before, a lot of spikes on the graph because of this switching occurring during the fermentation when your main carbon source is depleted. And this also demonstrates how it grows over time. So this one is complex medium. As you could see, it's OD 600. It grows way faster, but it reaches this point and doesn't grow really any further. Mineral medium, slow but steady. It's able to reach way higher cell densities also because of lower accumulation of byproducts, because they don't generate as much byproducts as in rich medium. And specific growth rate, yes, indeed, at the beginning is huge, but then it drops and doesn't grow in complex medium. In mineral medium, it's sort of steady. The same with dissolved oxygen consumption. The thick line here is complex medium, and it sort of reminds you the graph I showed before, when there was a lot of oscillation of oxygen consumption just because of many potential carbon sources. In mineral medium is very, very predictable and consistent, and actually it sort of reflects the exponential growth of the culture itself. So by looking at that, you see that complex media are not very robust and very variable, or the processes that are based on complex media. Meanwhile, switching to mineral media, you assure higher robustness of your process. It was also demonstrated by Thermo Fisher themselves when they took their Bacto CD Supreme medium, which is a chemically defined microbial medium, and compared lot-to-lot variability in terms of plasmid production, compared to very common terrific broth medium, which consists of yeast extract and a peptone, either animal or non-animal derived. And as you could see, this is actually a straight line with not much of variability. Here, again, it jumps as a goat in Tatar mountains, up and down. There is no really reproducibility here, and you cannot really predict. Imagine you are a producer of plasmid DNA, and you cannot predict how much you would produce tomorrow if you use the complex medium. What would you say to your client? How much would it cost? You cannot even make any business-based assumptions because you don't have a stable process. So even if it feels like a scientific problem, it's actually a business problem. And also mineral medium, because of this robustness of this process embedded, helps with actually scaling up. I used the same approach, the same script, automated script, combined with mineral medium in 3 different sizes of the bioreactors, 0.3, 1, and 3 liters. And even the traces, the fermentation traces, were nearly identical. There is a bit of a difference here because of different KLA or different bioreactor sizes. But the protein expression levels, total volumetric yield at the end, and total OD at the end was actually fairly similar. And it was just a copy-paste application, and that's it. The whole scale-up was done this way. Because, again, you could always write a script that self-adapts to the geometry of the vessel, to the range of agitation available, and to the range of aeration available. So now let's look at a use case. So during my tenure at Oxford University, we were developing new bioprocesses, or we were helping academic researchers to transit from the lab scale to the production scale. And I worked at Jenner Institute, which is well known because of their vaccine works. They produce a lot of vaccines, that some of them are recombinant protein based. And in order to increase the speed of vaccine manufacturing and development, we tried to adopt a process of transient expression in CHO cells. And the cornerstone of this process was flow-through electroporation. It all sounds good until it doesn't. And the process was potentially very advanced with one minor drawback. It had to use plenty of plasmid DNA for electroporation. The process of electroporation, especially of the CHO cells, wasn't really efficient, and in order to make it happen, you'd have to spend a lot on plasmid DNA from a commercial source, and it was fairly expensive, especially for academia. It was very expensive for academia, even if it's Oxford University, which is one of the best in the world and quite rich, but still, it doesn't directly translate to amount of money in a researcher's pocket. So we decided to develop our own platform that is capable of delivering gram equivalents of plasmids from benchtop bioreactor. We decided to move away from a brute force approach when you need 1 gram of DNA, and for this you will set it up in 200 liters of TB medium, and you'll get your 1 gram in the end. So we decided to go with intensification of the process. And that's how it looked in general. So you have two main, let's say, three main phases. One phase is the batch phase, when you start with a 15 to 20 grams per liter of glycerol, and you wait until the depletion of this glycerol. And you see this blue line, blue DO spike here. When you see that all the glycerol is depleted, you start your feeding in exponential mode until you get some OD of around 60. This is an arbitrary number based on capabilities of your fermenter system, because you are going to be limited by oxygen delivery, by heat transfer, by chilling capabilities of the system. Everything that we mentioned during the first presentation, what's different? Because the process is very intense in terms of heat and mass transfer and agitation, etc. So agitation reaches like, let's say, 2000s. If your bioreactor is capable of reaching 2000s, you'd get maximum. And at a certain point when you have enough OD, you switch the temperature from 30 degrees to 42, 41 degrees. And that uses one feature of PUC-derived origins of replication of plasmids. They increase their copy number from several hundreds to sometimes several thousands. So you could get up to 10 times more plasmids per cell. And because it's a very intense process for the cell, you cannot keep it up for longer. It was obtained empirically longer than 8-9 hours. Then the cells just cripple. They are not capable of maintaining any further growth and production. We also found out that based on the literature, what really works is to hold the cells at 24 to 26 degrees for an hour or two. That helps the cells to slightly recover from this high temperature shock and high plasmid production. And also, it repairs the plasmids that were produced. And despite of lower temperature, it actually increased the final yield by 10%, which is counterintuitive because the most of the accumulation of the plasmid is during the exponential phase at higher temperature. And then you just chill it and harvest, and that's all process. So obviously at that point when we started developing this platform, I already used mineral medium. I didn't use complex medium. And it was in-house medium made of 15 different components. And I was quite efficient at making this medium because it's very important how to mix the salts so that they don't precipitate, especially trace element solutions, heavy metals, they don't behave nicely. And I was quite happy with actually getting quite good results. 250 to 160 milligrams per liter is not a small amount. Imagine that at the very beginning we had like 40 milligrams per liter at the final volumetric yield, and specific yield was around 0.6 milligrams per OD per liter. And I was quite proud at getting 1 gram from the total volume we used to have. But then I bumped into Bacto CD medium. As we switched to Bacto CD medium, we saw at first that if we don't do anything else, it delivers the same result. But you don't have to spend days in making medium. You don't worry about doing a mistake, doing weighing, diluting, adding, sterilizing this medium. And also we saw a potential benefit in this medium comes with GMP certificate. So whenever we want to switch to go to the GMP suite and make it under GMP, we don't have to do any paperwork. With 15 components in your classic mineral medium, you'd do quite a lot of paperwork and you'd hate it. And then this initial spark of interest ignited, we had a conversation with field application scientists. They said, what if you'd try our beta product, which is Bacto CD feed? Obviously, and it was for free. Obviously, I agreed. Let's give it a go. And without changing anything, just using Bacto CD feed combined with Bacto CD Supreme medium, I got 60% more. And again, before that, I was making my own medium with some amino acids that I derived from the literature, from the metabolism and biosynthesis of DNA. It was fairly complex. I had to balance, for example, nitrogen sources, amino acids, and sulfur sources to reach to this 3.3, which I was very proud of because it was above 3. With this medium, it's just one component. You dilute it with water and add some glycerol, and you're done. Again, it comes with GMP certification, so whenever you want to tech transfer or scale it up later on, no barriers whatsoever. Everything is done for you by Thermo Fisher Scientific. It's more expensive, I agree, but as a well-paid lab scientist, my time is probably way more expensive than 13 pounds per liter of medium, because to prepare it, I'll spend 5 minutes instead of 2 days. So again, as I mentioned, this process was automated. And whenever I tried to automate something that is based on complex media, it wasn't as easy because it was less predictable, and computers are not smart enough to recognize the right point when to start feeding or when to switch phases, when to increase the temperature, all of that. And the mineral mediums helped a lot in providing a very powerful tool to automate any bioprocess. And that's probably for your information. If you really want to establish something like plasmid production, it gives you the theory behind the automation, because actually when the medium is well defined, the bioprocesses are also well defined and predictable, and you can actually hardwire it into your script and it will work every day the same. So this is actually, I'm not going to read it through. I hope you'll get your presentation pack later on and you could refer to it if and when it's necessary. Also, if not completely clear what's on this slide, on my third slide, you could contact me and we will go through it together. So, conclusions. I already told that. So, Bacto CD Supreme FPM along with Bacto CD feed provided very high plasmid yields in bioreactor settings, allowing to push it. In my 10-liter bioreactor, I used half of the volume at the end, less than half of the volume really. So if I would ever utilize the whole volume, it means within 2 days, I have 4 grams of plasmids from a benchtop bioreactor. And the price of a research grade of this material would be approximately $40,000 if you buy it from elsewhere. All you need, obviously, is to somehow process it. It will add some costs, but as you can imagine, you already are saving a lot of time, efforts, you have your robustness, and you're still getting that much of plasmids from a tabletop bioreactor. And even when compared to in-house mineral medium, which, by the way, you could probably make 10 times cheaper than Bacto CD if you will be able to prepare it right. And it's very important if. Still, Bacto CD Supreme will be quicker and simpler in terms of medium preparation. It will save you a lot of headache and time. because you don't have to worry that something didn't work and you don't know why it didn't work, whether because of the medium or whether because of something else. Here you can remove the medium from this equation completely. And we sterilized Bacto CD medium using a filter, but according to the notes, you could also sterilize it by autoclaving. We didn't do that because cold sterilization is actually better, in my opinion. It saves a lot of energy, it saves a lot of time. You don't have to book the autoclave suit to actually autoclave it. You don't have to wait until it's cooled down. And still, I think when you heat something to 120 degrees and then cool, there might be some changes, even though it wasn't shown, it wasn't demonstrated. Rather, the opposite was demonstrated. There is no difference between modes of sterilization. But I would rather be cautious on that. If I can avoid speaking to autoclave guys, I would avoid speaking to autoclave guys. And also, when and if you want to scale it up and run a GMP process to manufacture your drug, your vaccine, whatever, you can do it easily because you don't have to think too much about how to make your process GMP compliant. All you need is to write proper reports, SOPs, but the medium is there already before you started.